An important tool in classifying algae is the way in which the cells divide, and the way the organisms reproduce. This lecture is a quick review of cell division for both prokaryotic and eukaryotic cells. The short video of the desmid algae Microsterias shows one method of reproduction used by algae. On the left is a euglenid in the process of reproducing, and on the right is a colonial volvox before bursting and releasing the baby colonies into the universe. The reproductive strategies of different types of algae will be emphasized for much of the rest of this course. Prokaryote cells have a much simpler method of cell division than eukaryotes do. Prokaryotes grow and elongate. When they are large enough, they duplicate the DNA. The DNA is pulled to either end of the cell. The cell forms a septum, also called the Z-ring, where it will divide. The protein that makes the Z-ring is similar to the protein that makes the microtubules and flagella. Depending upon the species, cell division can occur in minutes or days. In E. coli, the lab rat of the prokaryote world, the generation time under ideal conditions is 18 minutes. Not all cells completely separate, as you can see from the filaments of Oscillatoria and the sheets of Merisnopedia. Prokaryotic cell division is called binary fission. Binary fission is asexual. This means that there is no genetic recombination during cell division. The DNA molecule replicates itself when bacterial reproduction takes place, and each daughter cell has exactly the same DNA as the parent. All descendants of a single prokaryotic cell are clones. Prokaryotes are able to mix up their DNA either by taking DNA fragments from the environment and incorporating them into their genome, or by conjugation, where two prokaryotic cells join and swap fragments of DNA. Eukaryotic cell division is much more complicated than prokaryotic cell division, and it has two essential but distinct parts: the division of the genome or nucleus and the division of the cytoplasm. The division of the genome is called mitosis. The division of the cytoplasm is called cytokinesis. In general, mitosis is followed by cytokinesis, but not always. Before we get too far into discussing mitosis, we need to learn some new words. Ploidy is the number of copies of the genome per cell. Is a chromosome present in one copy, two copies, or more copies? Haploid, denoted by small n, means the cell has one copy of each chromosome in the cell. Diploid, denoted by 2n, or twice small n, means the cell has two copies of each chromosome in the cell. Prokaryotes only have one copy of the genome per cell and are by definition haploid. Most eukaryotes have two copies of each chromosome in the cell and are diploid. The reproductive cycle of eukaryotes alternates between haploid and diploid stages in the cell cycle. Some eukaryotes spend most of their life as diploids, like you. Other eukaryotes spend most of their life as haploid, like some algae. And some more new words. Interphase is the period where the cell is not dividing, but is growing larger. Mitosis is the period where the duplicated genome is segregated into two new nuclei. Cytokinesis is the period where the cytoplasm divides into two distinct cells. Mitosis is divided into four phases: prophase, metaphase, anaphase, and telophase. During interphase, the cell grows in volume and produces all the proteins and other molecules needed for growth and survival. For our quick review of mitosis, we will use playing cards to represent chromosomes. We have jacks, 3s, and 9s. The red cards represent the maternal chromosomes, and the black cards represent the paternal copy of each chromosome. This is a diploid cell. The blue line represents the nuclear envelope, and the pink line represents the cell membrane. Near the end of interphase, the genome duplicates. The first phase of mitosis is prophase. During prophase, the chromosomes condense. You can think of it as loose yarn winding itself up into skeins. When the chromosomes condense, the centromeres become visible. The centromere is where the two daughter chromatids are joined. The centromeres are shown as green dots in the cartoon. In prophase, the nuclear envelope dissolves in some cases, and the mitotic spindle forms from microtubules. The second phase in mitosis is metaphase. During metaphase, the chromosomes line up along the metaphase plate or equator, and the centromeres are attached to the spindle by a protein complex called the kinetochore. Anaphase is the shortest stage during mitosis. During anaphase, the centromeres and kinetochores split or disintegrate, and the identical copies of each chromosome are pulled or migrate to opposite poles. This image has a microtubule stained in green and the DNA is stained purple. You can see how the spindle is pulling the two sets of chromosomes to opposite ends of the cell. The last phase of mitosis is telophase. This is where the chromosomes decondense, or you can think of it as the yarn unwinds, the spindle dissolves, and the nuclear envelope reforms. This gives rise to a short-lived single cell with two genetically identical nuclei. The last step in cell division is cytokinesis, or division of the cytoplasm. At the end of cytokinesis, the daughter cells are genetically identical to the parent cell they came from and genetically identical to each other. Mitosis does not shuffle the deck. It does not mix up the chromosomes. Mitosis makes two identical decks, or two identical genomes. One parent cell divides to make two genetically identical daughter cells. Remember, there is no genetic exchange during mitosis. What you start with is what you end up with, just more of it. Sometimes cytokinesis does not follow telophase at the end of mitosis. What results is a single cell with multiple nuclei called a cenocyte. As you can see in the cenocyte of Botryoidum granulatum on the right, there are 1, 2, 3, 4, 5 nuclei. The algae sailor's eyeball, Belonia ventricosa, is a species marine cenocyte found in tropical and subtropical oceans. It is a single cell with thousands of nuclei. It is quite possibly the largest unicellular organism on Earth.